All right. Hello, everybody. Welcome to this replacement lecture on the muscle topic of chapter 10. So while I'm visiting my mom, I hope you find time to listen to this lecture. Lecture 2 and 3 of chapter 10 will build on this foundation. And today we'll be talking about very basic aspects of mostly skeletal muscles and what makes them contract. You have received a good deal of information in the lab already. Here, we're going to expand on those internal structures of the muscle cell, and that's what we will cover today in this lecture. So let's get started. I will have some animations for you, some interesting pictures, and I need to get to the screen sharing part here. So let me see if I can start a screen sharing. So here is the beginning of this screen sharing. I'm trying to make this full screen. Here we go. So our interest in muscles in general is because they cause movement. They are a very unique group of tissues. And so their utility is visible or illustrated on this particular slide. In the lab, we'll be largely occupied by looking at skeletal muscles that produce body movement or stabilize body positions. If you want to regulate organ volumes, you want to release urine from a body, release from a fecal material. We have smooth muscle sphincters that has to be relaxed for defecation or urination to occur. And if you want to move urine or fecal matter out of the body, then you need to have a separate set of smooth muscles to cause urination and defecation. So moving urine, moving food or what's left of it at defecation includes smooth muscle. Moving blood includes the movement of cardiac muscle. Moving lymph, this is an indirect movement caused by skeletal muscles. And moving sperm, that's the movement caused by smooth muscle, and we'll talk about it in AMP2. Movement of air, this means inhalation. Mostly this is also related to skeletal muscle movement. In addition to all these movements, the contractions of skeletal muscle produce heat. In AMP2, we look at heat maintenance, the heat homeostasis in the body, and one reaction to hypothermia is shivering, is involuntary movement of skeletal muscle, and that's supposed to produce heat, counteract the hypothermia, the cooling of the body. When it comes to muscles, we have several expectations to fulfill everything that we require of muscles. So let's start with the most obvious, contractility. Muscles have to be able to shorten and create force. Most of today's lecture is about the internal structures in muscle cells that contribute to this. You have seen some illustration of this, the sarcomere, thick and thin filaments in lab already. In order to have contraction, you've seen in the lab that we need a neuromuscular junction. It was featured on that model. So we have to be able to excite this muscle into contraction. And this is the ability to respond to nervous system. The release of the chemicals, we'll see this in future lectures. So the muscle needs to be able to contract, needs to be able to be excited. Conductivity will also show up in today's lecture, we have to be able to move this excitation from the neuromuscular junction to the far ends of the muscle cells and muscles. And so we have to be able to conduct electrical signals. It requires a very unique structure. We didn't see this in lab. And other expectations are extensibility. Our muscles should withstand at least some degree of stretching, and elasticity, the ability to return to original shape after being stretched. We're going to look at structures inside a muscle cell that allows these two properties to happen. This is the last time, at least for a while, we mentioned cardiac and smooth muscle. The information is very similar to what we have studied in the lab and a lecture in a tissue chapter. So we'll be focusing on skeletal muscle in most of chapter 10. And these are mostly attached to bones, as you saw in the lab, some of them attached to skin. And skeletal muscle is striated and voluntary in nature. We mentioned this in the lab too. We have 700 plus skeletal muscles in our body. Each of those is a separate organ, and the cells have alternative names in addition to muscle cells. Sometimes these are called muscle fibers or myofibers. A little information about connective tissue. We mentioned both superficial and deep fascia in the lab. We have no way of demonstrating it. The most consequential connective tissue, however, is coming from this group of myciums. And we've seen on the lab model endomycium. I put on the board the perimycium and the epimycium. So let's see how these show up on the textbook illustration. So here you have a single muscle cell here. See the circled muscle fiber. This thin grayish structure here that wraps the muscle cell is the endomycium. You see here a fascicle or group of muscle cells. The wrapping around the fascicle or group is the perimycium. And the entire muscle you see here attached to a bone, the entire muscle is wrapped into the epimycium connective tissue. So this is kind of hard to imagine that really this is happening in muscle. So let's take a short side trip to a grocery store, and let's take a look at what we find that could be relevant to all these myciums. And I'm going to stop sharing here and go to another screen share. And this is what I wanted to show you. So I hope you can see this big piece of beef. Take a look at the epimycium. This confines the entire muscle, and sometimes fat accumulates in this connective tissue wrapping. So take a look at this. Entire muscle is surrounded by the epimycium. Here's another muscle with its own epimycium, and you see the sharp demarcation between the reddish muscle and the fat. Now, take a look inside. The fascicles of the muscle also accumulate fat. We will call this marbling of the beef, but you can see individual fascicles. Some of them are smaller, some of them are bigger. And so this fat accumulation magnifies these perimyciums. So marbling of the beef is nothing else but the fascicles showing up separated from each other by fat. So this is the visible effect of the epi and perimysiums. The endomysiums are not visible at this magnification anyway on this chunk of beef. I'm going to return to the lecture slides here. And so after these connective tissues, the endo, peri, and epimysiums, we have to mention the large visible structures they form, which are tendons. Aponeurosis, the epicranial aponeurosis we saw in the lab, is a flat tendon, and tendons are composed of the merging of endo, peri, and epimysiums. Very important portion of entire muscles, because only tendons and aponeurosis can grab onto bones. So if muscles want to move bones, they need this middleman, the tendon or the aponeurosis, which are made of connective tissue. We have to mention that skeletal muscles are highly active organs. They use a lot of ATP energy, so they need plenty of oxygen, nutrient supply. So we have an artery and 2 veins. We also need a supply from a nervous system. So to the neuromuscular junction, we have nerve fibers coming to each muscle, making each muscle a separate organ. So let's take a little closer look at the muscle cells or myofibers. This illustration looks just a little different from a model that we had in the lab. We'll see a few additional parts here in lecture. So muscle cells are 5 to 8, skeletal muscle cells are 5 to 8 inches long and have a thickness of a human hair. But even this giant cell is surrounded by sarcolemma, plasma membrane. And inside the sarcoplasm, we see these red-colored myofibrils. You saw this on the model too. What we didn't mention in the lab is that the reddish color of muscle is coming from myoglobin. It's a protein that can stash away a little bit of oxygen inside a muscle cell. We also find this molecule glycogen. We saw this molecule in chapter 2. This is a polymer of glucose. This stores energy for the muscle cell. So when we look at the plasma membrane, unfortunately, these structures weren't modeled on our lab model, but take a look at the true look of the sarcolemma. In addition to the smooth surface, there are invagination sinkholes, if you will, tunnels coming from the plasma membrane, from the surface, all the way deep into the muscle cell. These structures, these tunnels, are called transverse tubules. They are transverse because they go all the way across the entire cell. They are filled with extracellular fluid from the outside, and they have a tremendous importance taking muscle action potentials to all parts of the muscle cell. So I'm going to switch to a different image, shows you just a snapshot of an animation. And let me magnify this. So here we see a sarcolemma here in yellow. And here is how the excitation, that potential electrical charge, is going inside the muscle cell. These roundish structures are all transverse tubules. And so from the surface, excitation, action potential can go deep inside the muscle cells. So these are necessary structures to excite the muscle cell to signal that it's time to contract. Let me go back to the lecture slides here. And so in addition to the transverse tubules, the illustration in your textbook also shows mitochondria. Mitochondria produce the ATP, adenosine triphosphate, that the muscle cells and the myofibrils will be using. So they are strategically positioned all across the muscle cell. Muscle cells also have sarcoplasmic reticulum. This is strongly modified from normal cells. This sarcoplasmic reticulum, which is the endoplasmic reticulum, is turned into a calcium pantry. So we have calcium in solution filling these brownish tube-like structures, the sarcoplasmic reticulum. As we'll see in the second lecture, calcium will be an important chemical signal inside the muscle cell. So sarcoplasmic reticulum stashes away a large amount of calcium for the time when the muscle needs to start contraction. And so here are the myofibrils. We mentioned in the lab too that the secret of contraction is inside the myofibrils. Here you see one myofibril pulled out, and you can already see, I hope you remember this from the lab, the thick and thin filaments. A sarcomere is pictured here. We'll be immersing ourselves into this topic a lot deeper in a minute. This is just a little review if you want to test yourself about the parts, components of the muscle cell. So let's focus on the myofibrils. Here is one of them separated from the rest of the muscle. And you can see the rod nature of this. And you can also see the small parts, the sarcomeres that are located back to back, tens of thousands times inside the myofibril. And this unit is the sarcomere. The borders are the Z lines. Inside the sarcomere, in the middle, there's an M line. And we are seeing now the thick filaments anchored on the M line, and we see the thin filaments anchored on the Z line. This long parallel arrangement of the filaments is essential for the skeletal muscle to contract. Here is a little more zoomed in look. So when we take a look at the proteins making up the sarcomere, we are seeing now some of the proteins of the thick filament here and some of the proteins of the thin filaments. A bending pattern is created. When you take a look at this portion here, the bending pattern is forming a light or I bend, and towards the middle of the sarcomere, where the thick and thin filaments are found overlapping, we find a much darker band. This is the A band. And we mentioned this in the lab, the alternating A and I band provide the structural basis for striation. We mentioned this in the lab too, that these components of the muscle are proteins such as the Z lines, M lines, and also the proteins that make up the filament. So functionally, the proteins that participate directly in contraction are myosin and actin. We mentioned both of these in the lab. And then we have light switch kind of proteins that turn on and off muscle contraction. These are troponin and tropomyosin. There are also proteins listed here that we did not mention in the lab. These are provides for the integrity, elasticity, extensibility of the sarcomere and the muscle cell. And we'll be mentioning proteins here: titin, myomycin, nebulin, dystrophin. So let's say a few words about myosin. When you look at the bottom, you see a sarcomere and a thick filament in there. And now at this magnification, you can see the individual myosin protein molecules. When you look up here, you see that one myosin protein is having a long tail, and then it looks like a golf club at the other end. These are called the head groups or myosin heads. When you bundle a bunch of those golf clubs together, you get the thick filament. So this is why myosin is a major component of the thick filament. These head groups will be essential in causing the movement contraction when they grab onto the thin filament. When we look at the magnified version of thin filaments, we see several components. For contraction, the most important component is actin. Actin looks like these olive looking structures here. They are the handles that the myosin head groups grab onto. So they're really important to be located right here and available to the myosin head groups to grab on and pull the thin filaments to the middle of the sarcomere. We have troponin, tropomyosin arranged around the actin. Those are regulatory proteins, the troponin and tropomyosin. We'll come back to their function in the next lecture. So these thin filaments are held in place at the Z line or Z disc. And so the thin filaments are positioned so the thick filaments can grab onto them and pull the thin filaments towards the middle or M line. Here are some of the proteins that provide structural integrity to the sarcomere. Let's talk about titin. Titin means that it's gigantic. This protein starts from one Z line and goes all the way to the second Z line. This has to do with elasticity. If you stretch the titin protein, the spring-like portion of the titin protein will be stretched, and titin will pull the sarcomere back into its normal position. And when the sarcomere contracts, and the sarcomere is shortened in size, when you put pressure on the spring portion of the titin, it will spring back once the muscle contraction is over. The protein called myomycin, that is where the M line is found. So this blue chain-looking structure is made up of proteins. And the nebulin protein is just helping to maintain the alignment, the straight nature of the thin filament. Again, here's a little self-test if you would like to play with that part of the sarcomere, the myofibrils, and individual filaments and their proteins. We have one more structural protein that's worth mentioning, which is the dystrophin. So here's the sarcolemma plasma membrane. Dystrophin protein links the myofibril to the plasma membrane. So when this myofibril shortens, it can drag the plasma membrane with it. And this dystrophin makes sure that this entire structure is also connected to the endomysium. So the most frequent muscle-related disorder related to genetic mutation is muscular dystrophy. This is a protein, dystrophin, that is mutated in individual with muscular dystrophy, and muscle contraction, the force created by the sarcomeres, is not transmitted to the plasma membrane, the sarcolemma, or to the endomysium. And those muscles with these kind of defects cannot produce a very strong force. So here is the sliding filament model coming out from the arrangement of the thick and thin filaments. So here's the static version, and I'm going to show you an animation that you saw in the lab in a minute. So here is the relaxed muscle. Thick filaments and thin filaments overlap, and you can see the muscle, the myosin head groups grabbing onto the thin filaments. And as the myosin head groups keep pulling the thin filaments closer to the M line, we reach a point in a maximally contracted muscle where the thin filaments are pulled to the M line as close as possible. This is the shortest sarcomere. You can see, compare the distance of the sarcomere between the Z lines here. We have a shortening contraction coming from the shortening of the individual sarcomeres. So let me see if I can translate this into an animation. And let's see. Let me get out of here. Sorry about that. This is the animation that I want to show you. And just like in the lab, again, we will be observing the movement of the myosin head group, grabbing onto the thin filament. And when you drag the thin filaments closer to the middle, the Z lines will be dragged with it. So this attachment of thin filaments to the Z line, important. And let's start this animation. So you can see the purple thick filaments grabbing onto the green thin filaments, moving them closer to the middle. And this is the sliding filament model. Let me see if I can restart this. Right. So again, the myosin head groups of the thick filaments dragging the thin filaments and dragging the Z lines all the way until maximal contraction occurred. The sarcomere is shorter, and therefore we shorten the entire myofibril, we shorten the entire muscle cell, and we contracted the entire muscle itself. So I'm going to stop sharing here. So this was it for the first lecture. In the second lecture, we'll explore further details in the workings of the skeletal muscles. So lots of fun to be had. We have enough information for a third muscle lecture. Again, I hope you guys have a great weekend, and I hope to see you on Wednesday in lecture.